Hello, welcome everyone. Thank you so much for joining us today in our next edition in our MiDOG webinar series. I'm Dr. Janina Krumbeck. I'm the CEO of MiDOG Animal Diagnostics. And today's webinar is hosted by us at MiDOG here, as well as the SDVMA, the Southern California Veterinary Medical Association. We're really excited that you're here. Today's talk is going to be on gastric C. serpentis in snakes, and CE credit is available for everyone joining our webinar live today. You will see a QR code in the chat to download the certificate from there, or you can also download the PDF file directly from the chat too. If you have any questions, please make sure to put them in the chat. We will go over all of your questions towards the end of the webinar so Dr. Bogan doesn't get interrupted. Yeah, and that brings me to today's fabulous speaker, Dr. James Bogan. He's a board-certified reptile and amphibian specialist and is leading the voice in reptile health, conservation, and molecular diagnostics. He currently serves as the Chief Veterinary Officer at the Central Florida Zoo, and he oversees the Orianne Center of Indigo Snake Conservation, the only facility actively breeding the eastern indigo snakes. As a clinician scientist, Dr. Bogan has published extensively on reptile infectious diseases, gastrointestinal pathology, diagnostics, and reptile husbandry. His work includes pioneering efforts in C. serpentis diagnosis and treatment in snakes, as well as advancing molecular approaches to reptile medicine. And with that, thank you, James, so much for being here, and please take it away.
Thank you. That's the introduction. Wow, thank you. Let's see, go to share screen. How's that? Is that up? Great. So yeah, my name's James Bogan, and I'm just going to jump right into this. And I have another screen here just talking about gastric cryptosporidiosis in snakes. And just to get started, I'll go an overview over what Cryptosporidium is. And Cryptosporidium is a unicellular parasite of the Amphicomplexa nation. It used to be considered a coccidia. And recent molecular analysis has actually shown that it's more closely related to gregarines. These are parasites of invertebrates. So not related to the coccidians, more related to invertebrate parasites. If we look more closely at the phylogenetic tree of Cryptosporidium, there's over 38 species that have formally named, and there are 40, and actually some of the literature is close to 70 additional species that have been identified yet to be formally named. The vast majority of these infect mammals. Most of them infect the GI tract. And the species that we're really concerned with when we're talking about reptiles is C. serpentis, C. varani, C. testudinus, C. ducusmarki, and actually C. avium. There was a paper published several years back that some uromastics, a collection of uromastics, got really sick due to an infection of C. avium. But primarily when we're talking about crypto in squamates, we're looking at C. serpentis and C. varani. The prevalence of this is anywhere between 1 and 57%, depends on which population you're talking about, whether it's wild or captive, and depending on what tests that the investigators had used. Some tests are more sensitive than others. The physiology or the pathophysiology of this condition isn't really described, and it's just assumed that it is the same as the mammalian cryptosporidiosis pathophysiology, and that is a fecal oral route. But in mammals, like humans, you get a cholera-like diarrhea, anorexia, weight loss, abdominal discomfort, and the immunocompromised folks are more affected, whether you're very, very old, very young, or you have HIV, or you're undergoing chemotherapy, something along those lines. If your immune system is working fine, you spontaneously recover so long as you can be supported physiologically. You're not getting dehydrated or whatnot. Otherwise, you'll get over it. Your immune system takes care of this. Snakes will ingest infected oocysts through contaminated food or water, and inherently, Ophiophagous snakes or snakes that eat other snakes would be more at risk, you would think. But that's only if you're, if you have a snake in your collection where you have a king snake or a Drymarchon type of snake, and they are, but they aren't being fed snakes, then they're not any more risk than any other species. It's only if they're actually actively consuming other snakes. Interesting enough, these two species that infect squamates, C. serpentis and C. Orianne, have more organ tropism than they do species tropism. So it's more related to a specific organ than it is a species. So gastrotropic species C. serpentis, that infects snakes and lizards, but infects the stomach, where C. Orianne infects the intestines of lizards and snakes. Interesting, you see this photomicrograph, and let me see, get a pointer here. So you see all these little dots here, and these are all cryptoorganisms, and it is described as an epicellular parasite, or it's along the surface of this cell. These are epithelial cells of the GI tract. But these guys technically aren't epicellular. They actually occur within the phospholipid bilayer, the mucosal cells. Remember from Biology 101, you got that hydrophobic and hydrophilic double layer cell membrane. And the organism lives inside that membrane. So they're technically an intracellular extracytoplasmic parasite. And this is what makes treatment so difficult, because they're in this protected area. And here's a scanning electron photomicrograph showing just that. That's pretty cool. Anyways, we'll go over a life cycle here briefly, and it's pretty complicated. They have the option of reproducing either sexually or asexually. And so this will start with an ingested oocyst right here. This will release sporozoites, which infect the epithelial cells, and they'll convert into a trophozoite, which will form a type 1 muront. These type 1 murons will release type 1 merozoites, which reinfect the cell, either as the trophozoite in the beginning, or it'll develop a type 2 muront. These type 2 murons will develop type 2 merozoites, which reinfect the cell and start the sexual reproduction option as an undifferentiated gamete, and then it'll differentiate into a microgamete or a macrogamete, a boy or a girl. These microgametes release microgametes, which fertilize the macrogamete and develop a zygote. This zygote will release a thin-walled oocyst, which will reinfect the host, starting the whole process over again. Or it'll release a thick-walled oocyst, and it'll shed it into the environment and share the wealth with the world. So the pathophysiology in snakes for C. serpentis, this parasite will colonize the gastric mucosal cells, and it'll cause mucosal hypertrophy, and that'll lead to the wall of the stomach to thicken to the point where the lumen of the stomach narrows. Potential outcomes, you either have clinical disease, and we'll call that symptomatic. Now, technically, we shouldn't be using the word symptomatic in veterinary medicine, because symptom is something your patient is describing to you, and we have patients that don't communicate with us properly, so, or properly, like as a human would. So technically it's a clinical disease. Subclinical carriers are asymptomatic critters. And then you can possibly even have a transient infection, which would also not show any clinical signs. So the subclinical carrier, this animal will have parasites in their stomach cells, and it's not causing any obvious change. From the outside, you'd have no idea. The snakes are behaving normally, they're eating, they're reproducing, they just act normal, yet they're still shedding oocysts, and it's been documented that this can happen for over 20 years. Oh, frustrating. Disease snakes, you'll see lethargy, anorexia, weight loss, regurgitation. You might see a mid-body swelling, and some have been described to have a gastric intussusception. Now you see this photo here, this stomach, this snake's got a big bulge here. And this is what when I first were reading about this, way back when I was in vet school, and you hear about gastric cryptosporidiosis, mid-body swelling, this is what I picture. This photo is actually of a snake with a gastric adenocarcinoma. This is a tumor in the snake. This snake does not have crypto. Crypto snakes have this type of mid-body swelling. Very subtle. This is a dead snake. His head's off this way, tail's that way. And you can see here's where his gallbladder is. We get some staining from the bile. But this area right here, this is the degree of swelling we have. We see this black-tailed crebo up here. This here is the swelling. It's pretty subtle. At least I think it's subtle. So you don't always see mid-body swelling, or at least nothing that's really appreciable. So if we're looking at a necropsy specimen, I'm going to forewarn you, there's going to be quite a handful of necropsy photos in this presentation. But here we are with some pretty good changes. We get hypertrophy of the glands. So this is a snake, heads off this way, tails back this way. The body wall has been reflected. The stomach is pulled out. I slice the stomach open, and so I've opened it up like a I don't know, like a taco? I don't know. So here we go. You can see the thickened mucosal membranes. We have atrophy of the glandular cells. You can see the edema, excess mucus, a lot of inflammation. This is just icky. These changes impair digestion. And it can be so severe. Here's a snake that has passed from crypto. So again, here's the look at that stomach. Up here's the head, right? Back here's the tail. And I put this on here just to show you the severe emaciation. If you've ever necropsied a snake, this here is this particular animal's fat body. It is just withered away to basically nothing. This should be about 4 to 6 times this in diameter and white fat color. All right, diagnostics. So let's get on with some diagnostics here. How are you going to diagnose this? Well, first we need to determine what are we going to use as a sample. We could just use feces, and as a reminder, this is feces, this is not. This is urates. We can use vomitus or regurgitant, whatever they're coming up out of their mouth. We can run a cloacal swab, or we can do a little wash like an enema. We could do a stomach swab or a stomach wash. Now we want to do this 3 days after a meal. It's been shown that you're going to have the highest yield of any potential oocysts if you collect the sample 3 days after a meal. So we can see here, I like to use these swabs. And this here is, if you've ever used a uterine culture swab on a mare, it's the same instrument. This is about 70 centimeters long. This is a long swab that has got a nice protected sheath here. We could advance it. You can see I'm doing that here in this venomous species, protected in a clear acrylic tube here. I pass this swab down to the level of the stomach. And then once it's in the stomach, I can then pop the swab through this protective sheath and swab the contents of the stomach. It's a nice video here showing you that. Here's an eastern indigo snake being restrained. This snake is conscious, able to advance this swab all the way to the level of stomach. If you look here between Tony's hands, as I move the swab up and down, you can see loop, loop, loop. I'm swabbing the innards of the stomach. Stomach is quite far down in a snake. So if you're not using something that is that long, you're missing the stomach. And of course, we could do a stomach biopsy, again, 3 days after a meal. And the easiest way is to run an endoscope in, and here's a photo of that. Okay, now that we got the samples, what are we going to do with it? Well, if we talk to the biopsy, histologic exam, take this tissue, send it to a pathologist. They can then look for the organism as well as any pathologic changes or histologic changes associated with infection. We can, if we have any of the other methods we'd used, we can go ahead and just look right on the microscope. And we can see here, all these little circles, well, most of these circles, this one, this one, this one, this one, this one, these are Cryptosporidium oocysts. But some of these circles, like these back here, this is just garbage. They might be yeast buds. They might just be some ingested material. So pretty easy to miss if you're just looking on the microscope. So if we dry this slide and stain it with acid-fast stain, we can enhance the image. And all these red circles, these are Cryptosporidium oocysts. They don't all stain. Here's a couple here too. They're kind of ghosted out. But then that really differentiates between some of these yeasts and other miscellaneous crap that might be in there. You could also use direct fluorescent antibody testing. This really makes things pop, but you need a special chemical to make these fluoresce, as well as a specialized microscope. You can use an immunoassay. This is similar to what we use for our COVID tests, right? Same type of thing, what you're familiar with, heartworm tests, etc. And then, of course, PCR. Histologic exam is needed. It's the mainstay for proper diagnosis, especially if you're trying to diagnose a subclinical carrier. You need to have the histologic changes associated with disease seen, and not just presence of organism. Presence of organism does not equate to disease. Any of these tests I mentioned, with the exception of maybe a handful of PCRs, it's not specific to Cryptoserpentis. Many other cryptos, all the other cryptos, will cross-react with these tests. And so if you have a snake that's not showing any clinical signs of disease, and you run a stool sample and you're getting acid-fast little red circles, you don't know whether that's Cryptoserpentis, Cryptovarani, or you don't even know if it's Cryptomurus. It could be a parasite of the prey item that the snake is eating. So you have to send this sample out for DNA sequencing if you want to confirm that it is C. serpentis. Again, some exceptions would be like the probe hybridization qPCR that's designed specifically for C. serpentis. Now, there's no studies out there that compare all these tests against one another. So I scoured the literature and I found some detection thresholds that have been published. And if we look at these and I put them in order, acid-fast stain, you need to have 75,000 crypto-oocysts per ml to meet the threshold to be reliably picked up on acid-fast stain. ELISA, more sensitive, or the detection threshold, so to speak, is less. And so you need to have only 17,500. IFA, between 3 and 5,000. I put 4,000, you have to make it a round number, but it's between 3 and 5,000. Yet, if we look here, 40, not 40,000, 40 PCR, way more sensitive. And actually, qPCR, when you talk to the folks that do the molecular stuff, qPCR is even more sensitive than that. Now you couple this with the knowledge that subclinically infected snakes can shed as few as 400 oocysts per ml. If you're doing just these guys, you're missing it. You really should be doing PCR when you're screening. So I went ahead and did my own little study to look at this. I took some snakes, and since we know PCR was had a lower detection threshold and inherently qPCRs are more sensitive, I went ahead and did a qPCR that was specific for Cryptoserpentis, and I had 70 eastern indigo snakes where I anesthetized them and collected mucosal biopsies. I took this as my gold standard. If it was histologically normal, I called that normal. If it was histologic analysis that we had inflammation and a lot of body immune response to a pathogen, and there were cryptosporidial oocysts and organisms seen on histologic analysis, I call those positive. If I then I went ahead and took my mucosal biopsy and ran a qPCR on it and compared it to the histology. And that's what this first is here. I did a gastric lavage, and I did a gastric swab, and I did a cloacal swab, and ran them all at the same time. And then I put these in order of sensitivity because if you're like me, I want to know that the animal doesn't have the disease. I want the sensitivity. I don't want this false negatives. I want as few false negatives. And so the odds ratios are a real good way of looking at this. So if I could take a biopsy and did a qPCR of the biopsy, the odds ratio for a negative on a mucosal biopsy compared to the histologic review, greater than 501, that is a true negative. A stomach wash, same story, greater than 501. A stomach swab, greater than 501, so long as the animal was less than 163 centimeters long. Because if it was longer than that, then we're actually just swabbing the esophagus. Cloacal swab, only 72% sensitive. So a negative, oh yeah, maybe it's about not quite 10 to 1 odds that it's a true negative. But another way of looking at this is 1 out of every 4 positive snakes are being missed. So if you're just doing a cloacal swab, even if you're using the most sensitive test, you're missing 1 out of 4 snakes. Next, I went ahead and decided, let me look at this gastric wash and just compare some of these tests. You always hear these labs saying a PCR is not the same, our PCR is better and whatnot. So I collected a sample and I sent it to a bunch of labs. And the stomach wash, since the stomach wash was very sensitive, this was also a sample that was easily subdivided. And again, if we're looking at a probe hybridization qPCR, this is what you'll usually get at a university. Very good. These are excellent results. Now, here are some proprietary qPCRs that are run by a lab, one of the commercial labs. Actually does very well. I was surprised because this is a very inexpensive PCR. This here's another PCR. This is with the DNA sequencing. There was one of these 79 that actually tested false negative in this one here. So it wasn't as good as the qPCR. Interesting here, we had acid-fast stain. Depends if you centrifuge it or not off this stomach wash, if you're going to get decent results. So I don't need to read this all to you. You can see this right here, but interesting. This lab here ran a PCR. Not one of the positives popped positive, and a couple of the negatives tested false positive. So yes, not all PCRs are equal, and you need to understand the methodology that the lab is using when you send these off. If you'd like more information and really get into this, you can scan this QR code here. This paper is open access. This is published in the American Journal of Veterinary Research, and you can get all the details you'd like. So just kind of go over some endoscopy real quick. Here we go. So this is if you're running endoscope, you want to have your snake fully anesthetized, and you can use a pulse oximeter. I actually get a pulse oximeter run most of the time if I do this. And anymore, I'm not actually clipping on their face like this. I'm using a rectal probe, and I just stick it down into their esophagus, and I get some decent some readings there. Anyways, so our scope can run in. A lot of people are afraid of maybe using an endoscope in a snake. They feel that it looks like it's too big in diameter. But if you think about the prey item that these snakes are swallowing, this 0.9 centimeter endoscope is smaller in diameter than a mouse, and so there's no problem in sliding this all the way down. So we're going to look here at the stomach. The image on the left is the fundus, or the central part of the stomach, and the image on the right is the pyloric sphincter. So this is the back door of the stomach going into the small intestine. Normal. This is normal anatomy, normal mucosa. Here's a snake with some mild changes, and this is the quick note on the pathology report, but we just see some inflammation. The rugal folds aren't flattening out as well. Just looks angry. We see here some mild to moderate changes. You can see some edema developing in these mucosal cells. Kind of that cobblestone appearance. The rugal folds are much more thicker. And then some severe changes. There's no wonder that food can't pass through here. Look how thick these rugal folds are and how edematous these mucosal cells and hypertrophied these walls are. Here's another image of another snake with severe changes. All right, so what do I do for screening diagnostics? Well, it depends on what I'm looking at. If I have a snake who's been losing weight and it's been puking every meal that it's been fed for the last month, I might just take some of the expectorant, some of the vomitus, whatever it regurged up, and do a smear and do an acid-fast stain in-house. If I see red circles that are about 5 micrometers in diameter, I'm going to tell the owners that we're probably dealing with Cryptosporidiosis. Now, we'd want to get a biopsy to confirm that, but you can have a pretty good case there by doing that. Now, if I've got a snake that's got no clinical signs, and like say there's a snake entering a collection, I want to make sure he doesn't have it before he comes into the collection. That's where it gets a little more involved, right? So I'm going to want to either do my swab. This is what I really like to do. It's much quicker for most snakes, as long as they're long enough. Or you could do a stomach wash, and you could use, this red rubber feeding tube and a syringe with saline. 10 to 20 mLs per kilo is what you're going to administer, or you're going to use a stomach tube like this. Now, a quick image of a snake. So here's a cartoon diagram of what a snake looks like, and their stomach is about halfway down, right? So if the head is all the way here and the rump is there, this is where their stomach is. And if this snake was a little bit bigger, we'd have to weigh the pros and cons of the different ways of collecting this sample. If I were to take this stomach swab, it may not reach the stomach, which is here. Same holds true for this red rubber feeding tube. It's even shorter than that swab. And if I'm running a quote unquote stomach wash using this tube, you're actually just washing the esophagus. And I would not trust any negatives there. If it's positive, sure. Okay, then I'd like to look at that a little further, but a negative doesn't really mean anything if you're using this. So I like to use this hose here. This is a small canine stomach tube. This is what you would use if you are trying to decontaminate, poodle came in and ate a bunch of chocolate, you're doing, lavage its stomach. This is the tube you're using. And if you uncoil that, it's much longer. And if I pull this up here, yep, it's getting into the level of the stomach, and I'm going to have a better sample. Again, we're going to do these stomach samples 3 days after a meal. That's going to give you your highest yield. All right, turn the page here. We're going to talk about disinfection, some of the management for Cryptosporidiosis. And there was this paper published in 2010 that looked at disinfectant efficacy against Cryptosporidium, and it had determined, these researchers determined that a disinfectant needs to reduce the oocyst infectivity by 99.5% in order to be effective. If it wasn't 99.5% reduction in infectivity, it was not an effective disinfectant. So then I took that information and I created this chart here. This is a list of common disinfectants that have been tested against crypto, and I got the references on the right-hand side for where it was published. And if I'm going to draw a line right here, this is where effective, ineffective. Everything above this line is considered effective. Everything below this line is considered ineffective. And look at some of the stuff down here. Quaternary ammonium. I mean, this is your Rocal, your Kennesaw, Virkon, some of your phenolics, your all the alcohols, carbon monoxide, formalin. But what's effective? Hydrogen peroxide, greater than 6%. Some of these other ones here, you look farther up, like what the heck is oxyvirin? I didn't know. I looked into active ingredients. Oh, it's 25% hydrogen peroxide. No wonder why that works. Same thing with this compound. It's just basically concentrated peroxide. Now this company doesn't tell you what's in there, so who knows what's in there? It's probably peroxide. A lot of people talk about using ammonia, and ammonia will work, but if you reference the first paper that talks about ammonia for crypto disinfection, it's referring to ammonia gas, 5% ammonia gas for 18 hours. That's not really practical for most of us. Now, if you're looking at ammonia that's in liquid form, 6.64%, you need to have 24 hours of contact time for this. Now, a liquid ammonia that you buy, like you're going to go to Walmart and get ammonia, that's ammonium hydroxide, and that only has about 5 to 10% free ammonia in there. And some bottles right off the shelf contain as little as 2%. But assuming that you've got the regular between 5 and 10%, that probably would work undiluted for 24-hour contact time. That's pretty caustic. And if you're looking at like windshield wiper fluid, which is what this study here was using, if you're using something less at 2.5%, which is what some of the commercial bottles are already at, ineffective. Pretty close. We want 99.5, so that's close, but ineffective. These compounds have this chloro-M-creosol. If we look down here, we got 3% hydrogen peroxide, ineffective. Different contact times. Bleach, ineffective. Many people ask me about F10, and I don't know. I can't find a study anywhere on F10. That'd be a great project for somebody to get F10 and see how well it inactivates or how effective it is at inactivating cryptoosis. But if you look at the ingredient list, this is the only active ingredient that I see. There may be other proprietary that I'm not privy to, but that's the list, that's what I'm able to find. And this is the same active ingredient that's in these two items here. So I'm going to assume F10 is not effective. What about rescue? Well, if you buy the bottle, the concentrate bottle that you use for refills, that's at 4.5%. But if you're using the rescue wipes, those are only 0.5%. 0.5%, nope, that's not going to do it. The rescue concentrate, maybe. 4.5, I don't know. 3, no, but it's close. 6, yes. 4.5, I don't know, maybe. Other effective disinfectant techniques, of course, autoclave. Autoclave is effective. Gas sterilization, ethylene oxide is also effective. Hydrogen peroxide, the STERAD plasma sterilizer, that's also effective. And high temperatures are also effective. Now, moist heat is better than dry heat. When you're using your endoscope, you have to make sure that you are sterilizing between animals, otherwise you could be passing along. It's been shown that your cold sterilization, your glutaldehyde, whether it's the Alcock or the Alcock Plus, ineffective in inactivating crypto. You need to use ethylene oxide sterilization or the sterad sterilization. So what do I do if I'm looking for disinfection? You know, again, my endoscopes, I'm using the ethylene oxide. But if I'm looking at an enclosure, I'm going to thoroughly disinfect and clean it, you know, hot soap and water, get all the organic material out of there. And then I'm going to apply 6% hydrogen peroxide for 20 minutes. Now, I actually cheat. If I don't use 6%, I'll buy 20% hydrogen peroxide and dilute it to a third. So it's like 6.3, 6.5%. And the reason I do that is cost. So I just looked yesterday online here. If you're going to get the chemical grade hydrogen peroxide 6%, like from Thermo Fisher Scientific, you're going to spend about $1 a cc. That's a 100 cc bottle there, right here, 100 cc's for 90 bucks. But if I get this hydrogen peroxide from a janitorial supply store for cleaning, 20% hydrogen peroxide, a gallon is 20 bucks. And if I'm going to dilute this 3 ways, I'm basically getting 3 gallons of greater than 6% hydrogen peroxide. And that's a much more cost-effective way if I'm trying to disinfect a series of enclosures. What about treatment? There's not really any good treatments described. You know, you got to think about whether you're talking about a single animal versus a collection. Is this just a pet in someone's house? Is this a genetically valuable critter? Or is this, you know, you're talking about a ball python breeding facility and you have got one, you know, non-exotic morph that doesn't have intrinsic value. If we're talking about a business model here, then, you know, you're probably not going to take the risk of exposing everybody else in the collection. You might want to humanely euthanize to protect the rest of the collection. It also depends on whether you see clinical signs or not. You know, if you've got a singly housed animal and it's just someone's pet, they don't own any other snakes, and he's not showing any clinical signs, you know, we might look at some of these treatments I'm going to talk about that may lessen the severity, and some of these snakes can live for years without having any problems. Now, because Cryptosporidium is, again, not a coccidian, it doesn't respond to any of these coccidia medications. It's not even related to Giardia, so any of the medications you use for Giardia, it doesn't work for none of these. All these protozoal medications don't work. Some of the things that you might see in the literature, you might come across hyperimmune bovine colostrum. Now, this had some promise. This was investigated a lot in the 1990s, and this is where they would take a pregnant cow and they would inject it with crypto. The cow would develop antibodies against crypto, and then after the calf was born, they would strip the colostrum from mama cow, and that colostrum would be rich in antibodies against crypto. And there was some initial research that suggested it was very effective. And, you know, they looked at it in some Savannah monitors that had Crypto serpentis, seemed to help the majority of those. They did look at it in leopard geckos that had the intestinal crypto, and it did not help those guys. There was one study that looked at Indian star tortoises that had crypto. It didn't seem like it helped those guys, but there was a study in snakes that showed that if they didn't have severe crypto, it actually seemed to clear it. Now, the problem is this is not commercially available. Some of the naysayers for this were saying that, oh, these snakes are probably just got a very rich protein fluid and you gave them good nutritional support and they beat the infection. That's possible. Now, I say it's not commercially available. You may get on Amazon, you can look, and you can find HBC on Amazon, but this is HBC that's designed for scours. It's meant for calves that have E. coli infections. It is not specific for crypto, so that would not be something I'd be reaching for. Another drug that's commonly talked about is paramomycin. This might be your best bet. Paramomycin, when looked at initially, was so-so, but when they upped the dose to 360 mg per kg, there was a case report of a king cobra that was not showing clinical signs. They did 360 mg per kg twice a week for 6 weeks and tested negative for a year and a half later. Now, some of the tests were not as sensitive, so is it possible these are false negatives? It's hard to say, but he remained subclinical, and that was suggestive of some support here of efficacy. I hit my click button. Wait a minute. There we go. I did do a controlled study in indigo snakes using that same dose, and it did decrease the shedding, but it did not eliminate the parasite. When I challenged the snakes with a dose of dexamethasone to immunosuppress them, all the infected snakes started shedding again. So it does seem like it will decrease the shedding, but it won't get rid of it, at least at this dose for this duration. You might even consider higher doses. Now, this is an aminoglycoside, but it's poorly absorbed. So long as you're catching the disease early and you don't have gastric ulcers, then you should not be getting systemic side effects. But if you do have some GI ulceration, we have to worry about pancreatitis and nephrotoxicity. Another drug that's in the literature is nitazoxanide. Nitazoxanide is the only FDA-approved drug used for humans for crypto. And it really doesn't even work. Even the literature from the human side, it says it doesn't work. And they will prescribe this primarily in AIDS patients. And anymore, MDs will reach for correcting the AIDS or addressing the CD4 cells, the particular T cells, lymphocytes. And if they can get the CD4 T lymphocytes above 400, then crypto goes away. And so there was a couple studies that said if you combine this nitazoxanide with some other drugs like rifabutin and azithromycin, it was more successful in humans. They looked at this in leopard geckos, no success. I looked at it in indigos, and it didn't perform any better than placebo or control compound. Clofazimine, what is clofazimine? Well, this was a compound that was discovered in this study here by Love and colleagues, where they screened over 78,000 compounds in vitro, and they found that clofazimine was effective against C. parvum and C. hominis, the human versions. And it's already FDA approved for treatment of leprosy. So this was a great idea to repurpose a drug that's already out there. And they looked at it in immunocompromised mice, and it seemed to be effective. I looked at it in indigo snakes, and it was no better than control. And in fact, some of the snakes actually showed sign of toxicosis with this drug, so it may not be one I'd want to reach for. How about turmeric? Are you kidding me, turmeric? Yeah, there was actually a paper published in 2018 showing that curcumin, which is the active compound in turmeric, outperformed paramomycin in immunosuppressed mice. That's pretty cool. I tried it in indigo snakes. At least the doses that I used, it did not perform any better than control. Now there's this new drug that's a piperazine-based compound, which is parasiticidal. So instead of all these other drugs, which are static drugs, this is a cidal drug. It doesn't even have a name yet. It's called MMV 665917, catchy name. But I tried it in indigo snakes, and it didn't perform any better control. There's a newer version of this drug called SLU2633, and I'm just now finishing a study with that. This drug has cleared crypto in pigs and cows and immunosuppressed mice. It is also a cidal drug, and so we're waiting for the final PCRs on this to see where we're at, but so there might be some information to come here. Otherwise, you're looking at supportive care. And you might want to consider humane euthanasia. If we've got a snake that won't stop throwing up and is losing weight, you're just fighting the inevitable. Or if you're dealing with a collection, you want to prevent the spread, humane euthanasia. So how are we going to prevent? Big thing for prevention is proper husbandry. Correct the temperature, make sure they have correct humidity, correct diet, adequate hide boxes, basically stress-free. We want to try to get the cortisol levels low. We want to not immunosuppress these critters. And the best, that's what they do in humans. As long as your immune system's working fine, you get over it. So let's try to give the optimum husbandry to try to give them the best fighting chance. You want to prevent it in your collection, quarantine. You have to quarantine. 90 days minimum. There was a paper that had shown that the incubation period for Cryptosporidium in snakes is between 80 and 140 days. And that's based off of acid-fast staining of fecals. So it might be shorter than that. So I'd say a 90-day minimum. I like to do a PCR screen on entry. I'll do a gastric swab or a gastric wash collected 3 days after a meal. While they're in their quarantine for those 3 months, I monitor for signs of crypto. Before they leave, I repeat that screen again, just in case there was shipping fever, quote unquote shipping fever, and they were immunocompromised because of the stress of shipping. They may have tested negative initially, but they might start shedding oocysts a month or 2 into quarantine. And so you always want to check them again and don't release them from quarantine till you get those results back. Disinfection. Make sure that the enclosure and food bowls are cleaned real well, hot soapy water. You want to get all the organic debris off. Don't share cage furnishings and bowls between enclosures. Disinfect with whatever disinfectant of your choice for all the other cooties that could be out there. And then you want to repeat your disinfection with a 6% hydrogen peroxide for 20 minutes, minimum contact time. That way you'll help inactivate any cryptoocysts that might be still clinging on. So some considerations, historically you always hear C. serpentis is a death sentence. Not necessarily. I currently have about 15 to 20 snakes that have been testing positive for over 6 years that are still not showing any clinical signs. There are some of the literature does talk about some snakes over 20 years shedding cryptoorganisms, not showing any clinical signs. So if this is again, a snake that is in a single snake household, and it can be kept away from other snakes and other lizards, so you don't have to worry about spreading this to another squamate, then that's one way to just make sure they stay supported, good husbandry, etc. Again, immunosuppression is probably the primary player here. There's probably something in just being under human care. There's something that's being missed, whether it's adequate enclosure size, adequate diet, ventilation, etc. Some sort of stressors that's causing some immunosuppression. And probably the treatment behind this that's going to be successful is some sort of immune modulation or immune support. And with that, I'm happy to entertain any questions that may be floating out there. Thank you.
Awesome. Thank you so much, James. That was very great talk. We have a question here. It asks, does the 3 days after meal stand regardless of the size of the meal or the size of the snake? Or could that move to a shorter window? For example, with a small snake eating small items.
Yeah, this was it's coming from a study from the 1990s, and it's not. I don't think it matters between the size. Their typical meal. So if it's a smaller snake getting a smaller meal, and it was basically compared 3 days compared to 7 days. So you're going to have lower oocyst shedding 7 days after a meal compared to 3 days. Now, they did not do a study where they did it daily. So who knows, maybe 1 day is better, maybe 2 days is better, but between 3 and 7, 3 is better. And that's the only thing that's really out there. So we say 3 days.
Okay. Someone is asking if you will share the independent company that you used to do the PCR testing.
Yeah, so I've used on those tests there, it was University of Florida, for the PCRs was University of Florida, RAL, and VDL. The University of Florida was the one that was on the top. The second was RAL. Surprisingly, that one always gets a bad rap, but they actually did okay there, pretty good. But the VDL was what was on the very bottom.
Will this be available as recording? Yes, it will. We will put it on the MiDOG website, so you can receive an email later and you can watch this video anytime there. Next question, with a breed and release program, should we be concerned of snakes that test positive for C. varanae with no clinical signs?
I don't think there's enough known. I would not want to release snakes that are testing positive for C. serpentis or C. varanae into the wild unless it's already confirmed that those organisms are there in those released areas. Now, my personal belief is that if a snake, snakes in the wild get crypto, and I've collected snakes from the wild that have had crypto, but the ones that those snakes were always coming from an area that were, the area was under a lot of stress. There was, a lot of construction going on. Maybe it's a strip mine, or there was, reservoir development and a lot of bulldozer activity for years on end, which is probably creating some sort of noise pollution. The snakes will hear in very low frequencies, and large bulldozer type equipment will make noises in the low frequencies. So it should be in a range where snakes can hear. And I believe there's probably some chronic noise pollution that's probably affecting them, not to mention that they're being displaced and all the stress associated. And it's allowing pathogens to cause problems that normally they might just deal with for a few weeks and clear it, just like a mammal does.
Mm-hmm. Okay. Next question. Why do you soak for 20 minutes at 6% hydrogen peroxide versus 4 minutes at 20%?
There was the first paper that I had read that was earliest published was the 20% or I'm sorry, the 6% for 20 minutes. So that's what I do.
Okay. How can we access the recording? It will be on the MiDOG website. Next question. Would you recommend for screening hatchlings or very small snakes?
So what I do for hatchling is I wait till they're at least 3 months old, because in theory, they should not be exposed in the egg. If we're talking about an oviparous species, an egg-laying species, that hatchling should have been within inside the shell and would not have come in contact with this. The organism does not go into the oviduct as far as we know. And so the contamination of the egg could happen at the cloacal opening where the reproductive and digestive tracts merge. And so as the egg passes through the cloaca, it could pick up some oocysts on the surface of the egg. And so once the snake hatches, it could be exposed potentially to cryptoocysts. And if it got infected, it may take 90 days for it to start shedding. So I don't usually test my hatchlings until 3 months, 3 months of age.
Okay. And for everyone, the QR code that you see on the screen now, you can scan that to download your CE certificate directly there. For snakes and lizards, do you typically test for both serpentis and varanae?
Yeah, it depends what I'm doing. If if I have a snake or a lizard that's coming into a collection, so I'm at a zoo and I want to make sure there's no cooties in this critter coming in. I'll screen for all kind of pathogens. So I'll do a stomach wash looking for a Cryptoserpentis. I do a cloacal swab that's a pan crypto PCR, and then I'll DNA sequence the results looking for Serpentis or Varanae, or make sure it's not a passed through something else. And then of course other pathogens as well, whether it's nidovirus, serpentivirus, etc.
Is a low a low stress snake more likely to be asymptomatic when infected than an infected high stress snake, or does the difference just apply to the odds of infection?
It's a good question. I don't think we really know. Intuitively, it would make sense that a snake that is under higher stress is probably going to succumb to the disease more because it's going to be immunosuppressed, and so therefore would probably show clinical signs, where a snake that has less stress will probably be more immunocompetent and be less likely to show clinical signs. But I don't know that that is known 100%. That's just my gut.
Mhm. What types of habitats or cage furniture can truly be disinfected for reuse from a positive animal to a negative animal? Plastic versus glass versus fiber, others?
Yeah, probably. Yeah, anything that's porous, I'd be leery of. So, what I normally do, if there's any cage furnishing that isn't, super special, throw it away. Otherwise, if it's small enough and it'll fit into I could clean it and looks clean enough. Like, yeah, if this was a dinner plate, I'd probably eat off of it, and then that's clean enough to you. Then I'd run it through a ethylene oxide. But if it's
It's your own dinner plate? Oh gosh, okay.
well,
I think better safe than sorry. Throw it out. As far as stomach washes, is there a large concern for perforating past the stomach?
if say that again.
As far as stomach washes, is there a large concern for perforating past the stomach?
If you're gentle, there shouldn't be a concern. There's the stomach is pretty far down. Most species, it's 50% in. Now, those species that are ophiophagus or things that eat long snakes, the stomach might be a little bit more caudal. But if you've necropsied a snake, you kind of get an idea where the snake's stomach is located, and you want to make sure that your sampling item, whether it's the tube or the swab, is making it there. And I lubricate it well, and I'm just advancing with my finger, gripping only with my fingertips, and I don't grab it hard, just lightly grip with the fingertips so it easily passes. If I meet any resistance, I stop moving. But it should go 50% SBL.
Are snakes only carriers for C. varanae, or can C. varanae be detrimental for snakes?
Stevia rana can be detrimental. It does infect the intestinal layer, so it's not a stomach parasite. It is an intestinal parasite, and it can cause enteritis and diarrhea in snakes. So it's not just the varanae part is misleading. You want to think it's just going to be in monitor lizards, but it is in all squamates, snakes included.
Okay. Someone says, thank you, great talk. Do you think that the clinical state of the infection is more a factor of captivity and stress? Or in other words, have you seen clinical state in wild populations?
Yes and yes. So I think it's a probably more related to stress. And like I was just mentioning earlier, I have seen it in wild-caught snakes. I was part of a study where we looked at wild snakes that were collected out of pristine habitats, and over 225 snakes were sampled, and none of them tested positive. When I looked at snakes that were collected from areas that were undergoing a lot of terraforming, I guess what you call it, where there, a lot of bulldozer action going on, about 50% of those snakes were testing positive right out of the wild.
Someone is asking if you could show the etiology slides again, because they came in a little late. I don't know, we have already
You
this.
snooze, you lose, too bad.
Okay, it will be in the recording. We make the recording available this week. Okay. Is there value in breeding cryptopositive snakes that have no symptoms? Is this a genetic strength that can be passed on?
So, yes, there could be value in that. So if you've got genetically valuable animals and you can keep them separated from crypto-negative populations, if they are not showing any clinical signs, I've got snakes where I'm actually breeding for an endangered or a threatened federally protected species. They've been testing positive. Some of these snakes have been testing positive for 6, 7, 8 years, still producing offspring, and the offspring are remaining disease-free. Now, I'm very rigorous in screening those guys before they move anywhere. They'll have a minimum of 3 PCRs before they leave my facility to go to a Head Start facility, and they'll have an additional 2 PCRs there. So they'll have a minimum of 5 PCR tests before they get released into the wild.
Okay. Yeah, we have a ton of questions here. We've never had this many. So, James, if you have to leave, I can stop. Otherwise, I'm happy to keep going. One person's asking about the CE certificate. If you can't scan the code, we can also email you the PDF. So that's no problem either. Okay, next question. Does the hydrogen peroxide need to be diluted to 6% or a higher concentration still effective for oocyte deactivation? I'm assuming it's a cost-effective move to dilute, but wanted to clarify.
Yep, cost effective.
If a client has a collection of many snakes and lizards with mediocre cross-contamination prevention, washing dishes in the sink altogether, for example, and one of the animals becomes clinically ill with crypto and is confirmed with PCR and subsequent biopsy, what would you do? It seems like we should assume all animals are at risk and potentially infected. Additionally, a negative test may be a false negative.
Yep, that's when you panic, and it's a financial nightmare and a logistic nightmare. So basically, you need to screen each one of those snakes, and ideally, you're going to want to get biopsies. So endoscope, get a biopsy. Now you could just do a stomach wash, and if you want to do a stomach wash or a stomach swab 3 days after a meal, every single animal. And I would do what I normally do is I want 3. And so, because those can be shed on an intermittent basis. So that can get quite costly doing that many PCRs, but that would be the way to go. Is screen them with a PCR off of a stomach sample collected 3 days after a meal.
In wild populations that are concentrated in habitats such as under cover boards, do you think prescribed fire may be an effective management tool to decrease oocyst transmission?
That's a good thought. Probably, because fire is 100% inactivation for the oocyst, so that's probably a good way. But I'm sure there's areas, like if you're talking about somewhere in the southeast, and you got a prescribed fire in a longleaf pine ecosystem, that fire is not getting into that gopher tortoise burrow, and so there could be oocysts still down 20 feet underground into that burrow where the snakes may be cohabitating.
Mhm. Have you heard much about UVC sterilization?
There is there are plenty of papers that describe that, and most of that is centered around sewage treatment plants, because in developing countries where crypto can be a problem, there's a lot of UV discussion, and I've not been able to parcel out anything that's easy enough. Like, oh, I have an aquatic species and I got an aquarium and I have a UV sterilizer on there. I can't find that easy enough, and you know, I searched for a few hours and like, all right, I'm not that interested, and so I stopped looking. But there might be some information out there I'm just not aware of.
Yeah. Can breeding behavior and egg laying be considered a stress-causing time?
I would think so.
Okay. Can we get, oh, no, I already asked that. What's your opinion of the rapid antigen tests similar to the COVID-19 home tests that are available for reptile Cryptosporidius, usually from Chinese manufacturers? We have tried some recently on captive snakes and we are getting many positive results though it's very faint lines on the test. There are considerable doubt about the efficacy of these tests though.
Yeah, that was part of that paper that I shared on those blue screens halfway through the talk. And so I just used one US based company's um, test immunoassay, and it did pick up positives. It was not as sensitive as PCR, and actually not as sensitive as centrifuged acid fast stain off of a stomach wash. So it'll pick them up, but the problem is it's not species specific. So have to double check your positives. If you can take that same sample, set it in for PCR with DNA sequencing to see, is it C. serpentis? For all you know, it could be C. murus, again, a parasite of the prey item. Matter of fact, a study that we are just now finishing up with MiDOG, we've actually found some Cryptosporidium inside free-ranging eastern indigo snakes, but they are sequenced out as one was a chipmunk specific Cryptosporidium, and there was a couple of human Cryptosporidium that was just pass-through type quote unquote parasites and not actually parasites of the snake. So those crypto ELISA tests, those immunoassays, if they test positive, you need to speciate it out to make sure it's not a pseudoparasite.
Is it true that some snakes are more prone to infection by C. serpentis than others? And the examples given are Putriopis and Crotalinus. Sorry, I have no idea how to pronounce these.
Yeah, I don't know that that's true. I've seen it in Anthrophis, of course, Drymarchon. I've seen it in our Lampropeltis. So king snakes and in corn snakes and rat snakes, pine snakes, boa constrictors, ball pythons. Pretty much they all can get it. I don't know that there are any that are more resistant than others. I think some of the stuff that, some people may, and there may be some that are more resistant than others I'm just not aware of, but I think some of the stuff lately, our tests are way more sensitive than they used to be 20 years ago. And so the snakes that we're picking up now, like, oh, this one's got it, he must be more sensitive. That same species, all species probably had the same thing 20 years ago, but we were screening with acid fast and we were missing them. So, and so I guess
We
that's a
have
roundabout answer to say, I don't know.
which is fair. A couple of people are thanking you that it was a very informative and great talk. Someone is asking for part 2.
Oh my.
And then the last question, how much do you know about crypto and frogs? Does Varani or Septemtis affect frogs at all?
I No, Varana and Serpentis, I've not seen that or heard about either one of those in frogs. There is a gastrotropic species that I believe, I read about this 6 or 7 years ago, but I think the the frog that's used for research all the time. There was a gastrotropic species that was inside that frog. So yeah, but they have their own version, but I don't think Varani or Serpentis gets them.
Well, 2 more questions came in. James, are you still up for it?
Yeah, I'm okay.
Okay. Have you seen offspring from ovoviviparous snakes escape infection when born to a positive female?
I've not looked at any ovoviviparous snakes or viviparous snake species with it, so I don't know. But sure, I don't see why they couldn't escape it, but they are passing through the cloaca, so they could be exposed. And it depends on how, if mama defecated at the same time or close by and they crawled all through it, there you go. So
Oh. Any tips for doing gastric sample collection in small venomous snakes that are less than 15 centimeters?
yeah, I'll use those swabs. If the what I like about that swab is that it is it's stiff and it's not like trying to pass a rope inside a rope, so you can advance that as long as it's lubricated well. Less than 15 centimeters. I would probably, because I've done eyelash vipers conscious in their tube, so they're pretty small, but I took that swab outside of the protective sheath and just used it quote naked, I guess.
Okay, and that was the last question. Thank you so very much for giving us this wonderful talk, and thank you everyone for joining us. We will definitely have another seminar coming up soon, so please stay tuned on the MiDOG newsletter. If anyone had issues downloading the certificate, we'd be happy to email it to all the attendees. No problem there, and have a wonderful rest of your day. Thank you.
Thank you.
Bye.